Welcome, Jim.
Thanks,
Yeah.
Jerry. Good morning. It's good to see you again. Actually, I'll be talking about epigenetics. When I visited Iri a couple of years ago, we were talking with Kyung. He was asking me whether epigenetic trait would be good to study improving rice. I said probably yes, but my knowledge is very poor. So I studied quite a bit after that, and today I'll be focusing on epigenetic trait, how the epigenetic trait will control flowering time in rice. As you all know, rice and many crops has very extensive vegetative phase before they start flower. In rice in Korea, they put out on the field in mid-May and start flowering signal, so-called florigen, is made mid-July and then flower late August. So during this about 2 to 3 months period, flowering has to be suppressed. And there are many suppressors, and I think that major suppressor may be chromatin remodeling factor. So let's go to epigenetic regulation. In general, there are two stages. One is happening at the DNA level. The other one is happening at the chromatin level, especially histone protein. Histone protein, and that there are 4 types of histone protein, as you all know, and the N-terminal N is heavily modified by phosphorylation, acetylation, methylation, and other modification. And actually, a scientist knows a lot more about methylations, especially in plant. So I'll be focusing on methylation today. If the histone protein, histone 3 or 4, or if they are methylated, it will become either activate or inactivated depending on which histone is modified. For example, if histone 4 or lysine 4 is methylated or lysine 36 is methylated, the chromatin become activated. So that area will be transcribed. Whereas if lysine 27 or lysine 9 is methylated, then it become closed form. Therefore, gene around that area will be inactivated. I began with so-called BIN3 gene because in Arabidopsis, BIN3 gene is known to be involved in flowering time control. And Rick Emerson at Wisconsin University, he's a good friend of mine working on BIN3. So we start to collaborate on BIN-like gene in rice, whether any of them is involved in flowering time. As you can see here, this blue are rice and red are Arabidopsis. This is BIN3. And there are 2 genes, BIL2, BIL4 is very closely related with the BIN3, and there are 2 additional genes. I'll be talking about this first. BIL2 has PhD motif. This is a very important structural component because this PhD motif is known to bind to histone tail based on crystal structure from mammalian system and yeast. So this BIL2 has PhD finger motif, suggesting this may be chromatin remodeling factor. We have 2 tDNA insertional mutant. Very luckily, it's easy to study for phenotype. This is null mutant because there's no expression in the mutant. Phenotype of the tDNA mutant line shows late flowering phenotype. On the short day, there is some delay in flowering. Long day, of course, there's a little bit longer, and the very same thing, indicating that this is constitutive flowering activator. In other words, they need for flowering induction, lack of tDNA insertion caused late flowering. we look a lot of gene involved in flowering pathway, and as you can see there, these genes, there's no differences, except LFL1 expression is highly increased. Therefore, this gene, LFL1, might be target over BIL2. We measured transcript level starting from 22 days after planting, and we sampled 2-day intervals until about the 3 weeks intervals. As you can see, the BIL2 expression, this potential chromatin remodeler expression is low, and at about 30 days, you can see very sharp increase, and then stay at very high level from 32 days. Whereas LFL1 expression is exactly opposite. In other words, expression was high, and then decreased sharply, and then stayed low. That support, again, that probably LFL1 is targetable this chromatin remodeler. In the mutant, BIL2 mutant, this LFL1 expression transcript level remain high. This is wild type, as I already shown you. LFL1 was published in 2008 that this is involved in flowering control, acting as a repressor, and they showed some EMSA experiment demonstrating that LFL1 is actually controlling EHD1, which is major flowering regulator. In our mutant, LFL1 expression, the downstream expression, target gene is strongly downregulated. In order to prove that BIL2 is chromatin remodeling factor, we needed to demonstrate this protein bind to histone 3. We did this work with Xiaofeng Chao in Beijing. What we did was that we made fusion protein. Actually, we had to cut some kind of a portion of this protein, 3'N, and then fused it with GST. And that fusion protein was mixed with histone oligomer, and then did a pull down assay, and then we showed that the histone is precipitating, and this Western blot confirmed that histone 3 binds to BIL2. If BIL2 function as a chromatin remodeler, and if this actually target LFL1, then we should be able to immunoprecipitate with antibody raised against the BIL2. So this is the result. You can see that this is the area of the LFL1 which dissected with about 8 regions, 5 in the promoter region, and 3 in the middle of the gene, including intron and exons. And you can see that BIL2 was enriched in the promoter region, not in other area. These blues, these are a control. This is just a control. So this supports that BIL2 actually binds to promoter region. EHD1 is, I told you, this downstream functioning downstream of LFL1. We did the same thing. You don't see the enrichment supporting that BIL2 actually binds with LFL1. We did another experiment to directly shows that this part was actually modified by a histone modification. Therefore, we used antibody against histone 3, lysine 27 trimethylation antibody, and then we will be able to show the same kind of enrichment using this antibody. Therefore, we think that BIL2 mediate histone 3, lysine 27 trimethylation in this promoter region. But how this function in chromatin remodeling, and that is very interesting point. Actually, there are several kinds of chromatin remodeling complex, and this is one of the well-studied chromatin complex called polychrome group 2 complex, which is consisted of 4 core protein, as you can see, P55, ESC, EG, and SUG12. In Arabidopsis, that is another name of this is EMF2B. And this core complex actually trimethylate, as I told you, histone 3, lysine 27, and trimethylation, condensing chromatin structure. This is known from mammalian system. And when we look in rice genome, there are 2 copies of EMF2-like gene. So we named it EMF2B and 2A. And when you mutate this gene, what we did was we tried to see whether BIL2 binds with the EMF2B. I'll tell you later why we did that. And you can see that this is co-immunopolarization analysis showing that BIL2 actually binds with the EMF2B. The reason we did this was that EMF2B mutation has exactly the same phenotype as BIL2 phenotype. In other words, it shows late flowering, undershot day, long day, and paddy field. Therefore, we are proposing this model. So this is LFL1 promoter. Only about 3 weeks, this gene is highly turned on, I told you. And after about 4 weeks, this gene has to be turned off in order to give flowering signal. So in order to do that, it will, first step will be PRC2 complex will recognize and this element, EG, is probably recognizing this the histone core, and next step will be BIL2. I told you BIL2 is not actually a member of this PRC2 core complex, but this will bind with the EMF2B. And then I didn't tell you about this story. There's another member I told you. There are 4 copies in rice genome, and BIL1 is likely. We have some evidence we are working on. But BIL2, BIL1 is interacting and then pull this together. And then recruit polychrome group 1. And then after that, it will be condensated. This has to be solved further, but this is our model. It will recruit polychrome group 1, and then the gene will be turned off. Of course, some of this we needed to further do more experiment. So in flower network, this is kind of known. I didn't put all of them, but as you can see that phytochrome and GI is the photoreceptor controlling a couple of genes, including GHD7 and HD1. Those are, I also mentioned FL1. Those are actually all repressor. And polychrome group complex, PRC2 complex, together with the BIL2, suppress LFL1 to turn on this gene expression. Okay, I'll switch to next chromatin remodeling protein. Chromatin remodeling, I told you, major modification is by histone methylation. So the histone will be either methylate or demethylated. There are 2 types of methyltransferase. I mentioned you, one is PRC2 complex. This is another complex, tritorex complex. There are 2 types. And a third type of chromatin remodeling proteins is removing methyl group away from this. In other words, actually demethylase. There's poster in this session about this, if you are interested, so-called Jumanji group. I'm not going to talk about Jumanji group, but I'll be talking about another methylase called tritorex group. There are 10 members of tritorex groups, in rice. And in Arabidopsis, ATX1, they report that it's involved in flowering time. And this gene, we named the OS-TRX1 is most homologous with this Arabidopsis gene, which is involved in flowering. So we start from this one. That's TDNA insertion. And this encode for protein which has PhD domain. I told you that PhD domain is necessary to recognize histone molecule. And there's set domain which is involved in methyltransferase. Knockout mutation shows flowering, delayed flowering, similar to BIL2. However, if you see over here, in short day, there's very little differences in controlling flowering time, and very specifically delay flowering under the long day, indicating this TRX1 target different gene, not RFL1 I mentioned to you. We did methyltransferase activity. I wouldn't go detail. I wrote down over here if you're interested in. We basically made this because it's already know that set domain contain methyltransferase activity. We subclone this region out, fuse to MVP, and then this fusion protein is expressed in E. coli and incubate with histone. And then you look for with the radioactive material. You can see a methylation of a histone over here. I told you that TRX1 mutant flowered late. Therefore, target is likely the gene involved in under the long day condition. If you see around, first thing you can see is likely GHD7 because CIPA is very well-known paper that mutant shows very strong flowering phenotype under the long day. So it is likely this is target, or directly or indirectly. As expected, if we look for expression level, all other genes, there's no differences except the GHD7. EHD3 it was published a couple of years ago by Janos group, and this upstream gene of GHD7, there's no differences. Therefore, that will be working over here or maybe independently working. As you expected, GH7 expression remain high in this mutant, starting from 22 days. Therefore, we think that the GH7 is controlled by TRX1 and EHD3. Let me go back. Since they are working upstream of this, one possibility that they may be interact each other because TRX1, like BIL2, does not function by itself. It has to have a partner. So what will be the partner? Easiest thing is, what about EHD3 itself? So we made the fusion, TRX1 H8 fusion, EH3 big fusion, and then did co-IP experiment in right first. It confirmed that actually they bind together. We tried to see which domain is involved in the interaction, and we divided this area. As you can see, this PhD domain is involved in interaction with the EHD3. If you have set domain, interaction does not occur. So somehow we have to take out PhD3. In fact, EHD3 protein has 3 PhD domain, and TRX1 has 2 PhD domain. Either one of this PhD domain or both of them may be involved in interaction. We have to study further. So working model is that TRX1 has methyltransferase activity, but does not recognize DNA by itself. So there has to have some DNA binding partner, Also, there will be chromatin remodeling partner. We do not know whether EH3 function as a chromatin remodeling factor or DNA binding partner. It has to be resolved. This will be my last part of the talk. Another interesting gene emerging, Brett Meyer gave a talk yesterday, is that what about role of RNA, microRNA, for example. And we search through, there are about 600 microRNA genes in rice. We search through, and microRNA 72 I'll be talking about today. If you look for expression pattern of microRNA 172 throughout the day, it is northern blot, and end of the day, messenger RNA, the microRNA is accumulated. And we look for target, and there are 4 genes is likely target of microRNA 172. Therefore, we look through mutant of this one, independent mutant. None of them shows any phenotype. Not surprising because they may be function redundantly. This is the data. We see no or difference in flowering time. Therefore, we had to go for overexpression strategy. We overexpressed it. There are 4. I'm showing only 2 of them. It doesn't matter which one you overexpress. If you overexpress this AP2 family gene, then it flower late, indicating that AP2 is repressive flowering. If you see this AP2 gene, there is the sequence at the transcript level, and this is microRNA 172. There is target sequence. If you mutate the target sequence, we made a 4 base alteration. We named the 4M. And if we overexpress this one, it will never flower. So that meaning this is strongly repressor. So supporting that AP2 gene is target of microRNA 172. Try to find out what is the target of this AP2. We made a double construct between overexpression of this AP2 overexpressor and EHD1, which is working very downstream of flowering signal. And the phenotype is early flowering, in other words, follow EHD1, indicating that EHD1 function downstream of AP2. So this is our hypothesis. During vegetative stage, microRNA 156, I didn't mention about it, play a major role to keep vegetative stage. But as plant get old, the microRNA 172 expression increase, that suppress eventually AP2 expression, which acting as a repressor of EHD1. Therefore, this induced flowering. Let me put all this story together in one slide. Spring, early summer, for 2 months or so, depending on the cultivar. Let the long day condition. So this long day condition will be possible by phytochrome. Signal goes to GI. We have some evidence about this, but I'm not showing today. And that will give signal to suppress this pathway. And there are additional suppressor, LFL1, GH7, AP2, all they function as a repressor. So therefore, it will not flower. So if you become late summer, say July, for example, then day length becomes shorter. First thing they needed to do is this HD1 become activator. The same protein become activator. We don't know exactly mechanism how same molecule become activator from a suppressor needed to be solved. Then there is, I didn't mention about this when we published many years ago. There is transcription factor, MAG15, 51, acting as actually short-day specific activator. Then I told you that BIL2, together with the polycom group, EMF2B, suppress LFL1, so this will be out. Then TRX1, together with EHD3 suppress this gene. We put question mark over here because this one actually acts as an activator, so it has to have something in between over here. Last, I told you that microRNA 172 suppress AP2. Therefore, this will be all suppressed. That will cause you flowering. So to summarize, I told you that there are several independent flowering signal pathways coordinately operate in rice, and EHD1 is center of various flowering signal pathway. BIL2 and TRX1 induce flowering by remodeling target chromatin. MicroRNA 172 induce flowering by suppressing AP2 genes. And those are my students who work on this. I already mentioned about Charles and Rick Emerson as collaborator. Thank you very much.
We have a few minutes for questions.
Thank you, Dr. Dan. And just you mentioned the PRC2 complex is mediate trimethylation of H3K27 is involved in flowering time control. So I'm wondering, besides the EMF2, is there any other members of this complex is involved?
We have not looked for all of them. Therefore, I cannot say that. I'm sure that definitely yes. Just to make the answer short, all BIL-like family, BIL 1, 2, 3, 4, all they are involved in flowering. What about TRX complex? We don't know. We are testing it. We have knockout mutant for most of them. We are testing it.
Other questions?
Antonio?
That was a very enlightening talk. And you may remember that epigenetics is one of the agenda that I think should be considered in the post-sequencing area. So I think one of the reasons why epigenetics is not considered, it's not yet recognized as an important aspect in understanding basically economically important traits in rice, is that no one has yet to present an idea on how to use these mechanisms, particularly in breeding. So, do you have, more concrete ideas on how we can really promote the utilization of epigenetic principles in addressing agriculturally important problems in rice, particularly?
Thank you for a very good question. If we, I think depending on how you think, if you touch some of this epigenetic marker, some of the marker may be very useful, and because it control not only one, but it control overall. I told you only one trait of this chromatin remodeler, but they have a lot of other phenotype, and some of the phenotype could be very beneficial to agronomic trait. For example, as Chipa mentioned, flowering time is not all of them. Many flowering time gene is associated with grain yield and grain architecture. It has to be coordinated, regulated. Who control this overall coordination? I don't think it will be just transcription factor. It has to be working upstream. It could be microRNA or could be a chromatin remodeler. And I don't think every chromatin remodeler will be beneficial, but some of them would show very beneficial phenotype. I hope I can present that data next year.
Okay. Thanks very much.
Thank you very much.
Really a lot of information.
Thank you.
Yeah.